Mutation is the changing of the structure of a gene, resulting in variant form that may be transmitted to subsequent generations, caused by the alteration of single base units in DNA, or the deletion, insertion, or rearrangement of larger sections of genes or chromosomes. Mutation is the action or process of mutating. It is important as the first step of evolution because it creates a new DNA sequence for a particular gene, creating a new allele. DNA changes in the cells that produce the next generation. Mutations are changes in the genetic sequence, and they are a main cause of diversity among organisms. These changes occur at many different levels, and they can have widely differing consequences. Genes is made up of DNA, and DNA is used to give instructions for the production of proteins in the process of protein synthesis. The important concept is that there needs to be a method of determining which gene will be turned off and which genes will be turned off, which called gene regulation. There are many ways that genes are regulated. In human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation like cells in all of your cells to determine which portions of DNA is used. There is very interesting way of regulating genes that can sometimes be challenging to visualize. A way that has been found in humans, but found in prokaryotes with few eukaryotes exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it usually made up of few genes that involve enzymes. Enzymes are proteins with the ability to break down or build up the substances that they act on. Key players of an operon, where we can see some gene regulation. First, RNA polymerase. It's a builder enzymes actually because RNA polymerase is an enzyme. In biology, a lot of enzymes and in ASE are enzymes. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis, which means to make proteins, enzymes in this case. The thing about RNA polymerase, it gets a little confusing for RNA polymerase without somewhere to bind. Well, RNA polymerase needs promoter, where promoter is a sequence of DNA where RNA polymerase can bind. There is this other sequence of DNA called an operator. The operator is the part of DNA where something called a repressor can bind. The big bad repressor, if bound to the operator, it will block RNA polymerase, and RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins.